I'll be presenting today my topic on investigating the role of respiration in plant salinity tolerance by analyzing mitochondrial proteins from wheat and a salinity tolerant amphiploid. The idea behind carrying out this investigation was to identify the genes and physiological mechanisms that are specifically present in the tolerant plants but absent in the sensitive lines. These favorable alleles can subsequently be incorporated into elite agronomic lines through marker-assisted selection or genetic transformation. To the method, the methodology used for this experiment was 2D dye and peptide mass spectrometry and immunoblotting. 2D dye, the followed by protein precipitation, was conducted with all labeling, separation, and scanning. Side-eye was used for labeling. CY2 was used to label the internal standard, and CY3 and CY5 were randomly allocated to the protein sample. Gel images were statistically analyzed using the D-Cider program V6 GE Healthcare and mass spectrometry. The process aims to produce a collection of peptide fragment ions that differ in mass by a single amino acid, allowing one to read the amino acid sequence of the precursor peptide. Mouse number of matched peptides and coverage data are taken from Mascot search engine. Breakdown products were assigned to spots that are presented at lower molecular weights compared to a reference map of the wheat mitochondrial proteome. Coming to the section, we can see that in table 1, the rates of several physiological processes were measured in 7-week-old plants where the salt-treated plants had undergone long-term 200 millimolar NaCl treatment. Here, G represents genotypic effect, T represents treatment effect, and G into T interaction effect, and lower p values are indicated by more asterisk marks. The impact was that lower percent of shoot biomass elicited by salt treatment in both genotypes were expressed, higher salt tolerance in AMP, statistically root biomass, shoot biomass, and leaf elongation rate all exhibited statistically significant genotype into treatment effect according to the ANOVA test. In table able to differentiate the short-term adjustments induced to cope with onset of stress from long-term impacts of NaCl. In 7-week-old plants, salinity induced lower rates of respiratory O2 consumption, perhaps linked to slower provision of substrate. Root respiratory O2 consumption increased under salinity stress in both genotypes, reflecting energetic demands of ion exclusion. AMP leaves exhibited higher rates of respiratory CO2 efflux compared to CS under both control and salt treatment. In 5-day-old plants, both genotype exhibited increase of respiratory O2 consumption under salinity treatment compared to control. Interestingly, CS increased this parameter to a greater extent than AMP. Genotypic difference in salt-induced respiratory increases in CS. Coming to figure 1, mitochondrial isolated from shoots of salt-treated plants showed higher rates of maximal alternative oxidase activity in figure A for highlighting prominence of AOX capacity in shoot mitochondrial following salt treatment. In figure B, the measurement in this study did not detect higher AOX activity in mitochondria isolated from salt-treated root tissues. Whereas in figure 2, we can see the third methodology used, immunoblotting, that is, anti-AX antibody was used as first antibody in immunoblotting technique, and POD labeled anti-rabbit secondary antibody was used. Chemiluminescence was visualized using an Amersham image quant RT-ECL camera, GE Healthcare, and band intensity was quantified using Amersham image quant TL software, GE Healthcare. In figure 2, salinity treatment, role of AOX under salinity treatment using immunoblotting was seen, where the salinity treatment induced higher abundance of AOX protein in isolated shoot mitochondria in both genotypes with AMP containing higher baseline abundance of the protein. Immunoplot against AOX in isolated root mitochondria sample gave an indication that salinity treatment induced lower abundance in this protein in CES. Figure 3 and 4. Figure 3 and 4 are the images of Deitch, that is representing 2D Deitch images where the marked circle CY3 labeled shows green colored spot CS control treatment and CY labeled red colored spot shows protein from AMP salinity treatment. In this, we can see in this figures we can see that in shoot mitochondrial 2D Deitch comparison, protein spots derived from the highly abundant photorespiratory SHMT enzyme presented as a train of spots in mitochondria isolated from AMP. The basic end of this train contained a highly abundant protein spot that was dramatically less abundant in CS. In figures 3 to from table 3 to 8, we can see that significant differences in the treatment of tables 3 to 8. Various combination of physiological, biochemical and proteomic analysis was performed to explore the mitochondrial and respiratory responses to salinity in these two genotypes CS and MP. So the tables thus shows the significant differences according to treatment effect, that is P is less than 0.05 were evident in 55 mitochondrial protein spots deemed significant by the root comparison, but only 15 mitochondrial protein spots deemed significant in the shoot comparison. Highly significant genotypic effects were more numerous in the shoot mitochondria comparison to 7 protein spots exhibited P less than 0.001 for the genotypic effect compared to only 4 protein spots with this degree of significance in root data set. Thus, higher abundance of manganese, superoxide dismutase, serine hydroxymethyltransferase, acetonitase, malate dehydrogenase, and beta-cyanoalanine synthase compared to CS was observed for physiological and biochemical proteomics. This 3 to 8 physiological and chemical parameters being performed. Thus, the critical analysis author's aim was quite promising on investigating plant salinity tolerance. Convincing results with ample data collected. Indeed, this paper can guide crop improvement programs during catastrophic loss of crop productivity, climatic change in condition, also in case of widespread famine. References, and thank you for your time. Any questions?